Thanks, Leon, for the introduction, and thank you all for coming. This project has been a collaboration between Patrick Doyle's lab at MIT and Frank Slack's lab at BIDMC. Imagine you take a picture and your camera averages all the pixels in the image. You probably won't be able to see too much, but this is kind of how a traditional bulk sample bioassay works when measuring from tissue. You average over complex heterogeneous tissue and you get one number. People have been trying to get more information out of these tissue sections by doing single cell assays where they assay individual cells throughout this tumor. But that's kind of like your camera shuffles the pixels in the image. You lose a lot of that contextual information for how these cells are interacting with one another. And we're interested in developing spatially resolved bioassays that retain that contextual spatial information. And we think this is important because tissue is heterogeneous and has a complex organizational structure. We think that spatially resolved bioassays can provide additional data to inform fundamental biomedical studies and physicians making treatment decisions. And one of the biomolecules we're interested in measuring are called microRNAs, and these are promising biomarkers for cancer. They play important regulatory roles in many different diseases, and there are microRNA-based diagnostics and therapeutics in development. Moreover, the microRNA research tools market and number of microRNA publications per year have both been growing over the past 2 decades. It turns out that making spatial measurements of microRNA can provide a lot more information about a particular patient's disease. What this study did is looked at 2 different compartments within a tissue. They looked at the stromal cells, which are connective tissue, and the tumor cells. And they found that low microRNA 21 in the stroma correlated with positive patient outcomes, whereas low microRNA 21 in the tumor cells correlated with negative patient outcomes. And we've seen similar studies with similar conclusions that where you're looking for microRNA within a tissue matters when you're making some of these predictions or diagnostic decisions. Now, all these studies have been done using the gold standard method for making spatially resolved microRNA measurements. This is called fluorescence in situ hybridization, or FISH. And the main disadvantage of this technique is that it can only measure 1 or 2 microRNAs at a time. And the reason why this is a key drawback is that it turns out multiplexing microRNAs, or measuring multiple microRNAs from the same sample at the same time, can provide a lot of additional diagnostic information that helps you pinpoint what a particular patient's disease. It's been shown that panels of microRNAs can differentiate between normal and cancer tissue in many different diseases. But it's also been shown that they can distinguish between benign and malignant tumors. And this is important because they can tell a physician whether a particular patient needs a particular treatment, if any treatment. Now, all of these assays have been done in the bulk sample bioassay format. So this is in a tube. And you can imagine all of the additional information that's lost if you take a tissue section and put it into one of these tubes. So we end up in a situation where we can either do a bulk sample bioassay, where we can get multiplexing with limited spatial resolution, or we can do a spatially resolved bioassay where we can get that spatial resolution, but we're limited on multiplexing. And so there's a need for a technology that can bridge this gap. And so that's where our technology comes in. We can perform spatially resolved and multiplexed measurements of microRNA from tissue. And importantly, we can fit into the pathology workflow. So that means starting from formalin-fixed paraffin-embedded tissue, the typical starting material for most histological assays, with no sample prep in a short 1-day assay. The way our assay works is we take a nanoliter well array. You can think of this as a densely packed grid of square wells. And we fill each of these wells with microRNA detecting hydrogels and lysis reagents. We can take this nanoliter well array and press it against a tissue section on a glass slide with no sample prep. And the lysis reagents within these wells will digest the tissue, extract the microRNA, which is then captured by our posts. On the next slide, I'll show you some data that we took using this nanoliter well array from this tissue section from a genetically engineered mouse model for non-small cell lung cancer. We identified 3 different tumor regions labeled 1, 2, and 3, and some normal adjacent to tumor tissue. So here on the left of your screen is that same tissue section, and I'm going to walk you through 4 different heat maps corresponding to 4 different microRNAs. So first, if we look at miR21, we can see the general outline of the tissue section relative to the background wells. We can also see some differences between the tumors and the normal adjacent tissue. But what we didn't expect is we also see differences between different tumors within the same mouse model. So we see that region 2 is overexpressing miR21 relative to region 3. If we look at LET7A, we see that regions 2 and regions 3 are expressing LET7A at similar levels, whereas miR19B is being overexpressed by region 3 relative to the rest of the tissue section. So this information wouldn't have been apparent just looking at the H&E stain or just using a singleplex microRNA FISH experiment. This information could change the way new therapies are developed and the way patients are treated. For example, since we know this patient or this mouse has 3 distinct tumor types, we might recommend a different therapy than we would to a patient that just has 1 tumor type. So we think that our spatially resolved and multiplex bioassay can provide a lot more information than a bulk sample bioassay or a singleplex spatially resolved bioassay. We think this can lead to new research tools and biomarker-based diagnostics, and may also be used by physicians to treat patients differently. So the team is looking forward to answering any questions you have in the Zoom breakout room. We're looking for general feedback and advice, collaborators, industrial partners, and potential investors. Thank you.